Okay, you guys, so now we're going to be talking about eukaryotic cell structures and functions, and I'm going to give you a look at something that might be a little intimidating. This is the cell parts and functions list that I gave you to do, and this is the back side of it, okay? I'm going to go through some of these functions as I look at both the plant cell and on the other side the animal cell, okay? And I'm going to start with the nucleus, and I love this picture of the nucleus here because it's showing like some of the parts that we need to be concerned with the nucleus. The nucleus, we know it's the control center of the cell, and it's surrounded by the nuclear envelope. I call it a nuclear membrane a lot of times because it is a membrane. But why they sometimes stress envelope is because unlike our cell membrane or the plasma membrane, the nuclear envelope is actually like a double membrane. Okay, like a double rainbow or something. It's not just a single phospholipid bilayer, it's a double phospholipid bilayer, so it's almost like 4 layers in a way. Okay, deep within the nucleus, we have the nucleolus, okay, and the nucleolus, its main function is to assemble ribosomes. It's making the ribosomes, okay? Going back, sorry, the nuclear envelope is going to regulate what goes in and out. And the nuclear envelope is obviously spotted by little pores along there. And that's very important because RNA can like kind of scoot in and out of the nucleus, get made by the DNA inside there and scoot out to where it's going to be read by the ribosomes out in the cytoplasm. Okay, and chromatin. Chromatin is the genetic material. When we say chromatin, it's in the form of, like, I almost want to bring out my slinky and kind of show you that, like, when we talk about chromatin, it's as if the DNA is all stretched out like a slinky. And you can't quite see it so well, so we just say, oh, it's all chromatin in there. But when, and that's during interphase, but when a cell gets ready to divide, we know that that like slinky can like scrunch up, and that's what happens to the chromatin. It scrunches up and becomes these very visible chromosomes, okay? And that's going to be important a little bit later on, okay? So inside the nucleus, we have the nucleolus and the chromatin, and I want to add also the nucleoplasm. Nucleoplasm. I know this is going to leak to the other side of the page, but we're going to have to deal with that. And the nucleoplasm is just like the fluid that's inside there that is suspending the organelles inside there, okay? Moving on, and let's take a quick look to see how much it bled through. But anyway, we can see the nucleus in a plant cell as well. By the way, animal cell, plant cell, okay? And they both have a nucleus. So let's leave on here. And let's go on to the ribosomes, okay? And if we find the ribosomes in the plant cell, let's see where we can find them. Okay, and they're not labeled so much there, so we'll scooch over to this side. The ribosomes, okay? The ribosomes you're going to find in like different kind of areas, and I don't think, yeah, there they are. Okay, so ribosomes right here. Ribosomes can be spotted all along like rough ER, right, where you'd see these tiny little dots that are all like kind of scattered within here, right? But you won't find them on smooth ER, okay? But ribosomes can also be free floating out in the cytoplasm, like way out here or something like that, okay? This diagram doesn't show too well where they're pointing to, but hopefully you get the idea. Ribosomes are the site of protein synthesis. Boom. They just are going to make the proteins. But it's important to say about those two ribosomes, the ones that are free, so if we talk about like free ribosomes, okay, and maybe these are pointing to some free ones, versus rough ER that have like these bound ribosomes. Free ribosomes are going to produce proteins that are going to be used actually within the cell. But ribosomes that are bound to the ER, those proteins that those ribosomes make are eventually going to go out of the cell. They're going to be secreted out of the cell. So free ribosomes in the cell, and it kind of like is almost like backwards. You would think free ribosomes, oh, they're free to go out. But no, free ribosomes make the protein that stays in the cell. Ribosomes that are on the ER make the proteins that are going to get secreted out of the cell. And it's convenient that they're located on the ER because then that protein can be scooched all around and secreted outside of the cell in its proper location. Okay, so those are the ribosomes. Next, let's go back to that ER. ER is like just often said like, oh, it has these ribosomes or it doesn't, and that's what anybody ever says about the ER. Or they say, oh, the ER, it transports proteins. But you know, there's so much more to the ER. And we're going to start with the rough endoplasmic reticulum. The rough endoplasmic reticulum is actually going to be packaging the protein that's made by the ribosomes into these little vesicles, and you can kind of see some of these vesicles, like here's one here, and these vesicles are going to be kind of entering into the Golgi apparatus. They're going to actually help sometimes form the Golgi apparatus, okay? So the proteins from the rough ER, okay, after the proteins are made by the ribosomes, they transport in the rough ER and they break off in these vesicles, which help to form the Golgi apparatus. And eventually those ribosomes that make the protein, that scooch it down the rough ER, that may form into the Golgi apparatus, are eventually going to get secreted out. Okay? So it's kind of like a little production line there. But what about the smooth ER? The smooth ER, all this stuff, it's not making protein, because it doesn't have ribosomes on it, to go out of the cell. Instead, the smooth ER, very, unsung hero here, is actually metabolizing carbohydrates. So like when you eat your carbs, it's like helping to break down the carbs. It's also it's synthesizing lipids, fats. Unfortunately, if you don't burn off all those carbs, it's going to synthesize them into fats. It's also synthesizing steroids, and it's also a site of detoxing in your body. And an interesting thing about smooth ER is if you actually look at the liver cells of somebody who's very healthy and does not take a lot of medications or, recreational drug use or alcohol abuse, you're not going to see much smooth ER inside the liver cells, okay? But you look at the liver of somebody who's an alcoholic or somebody who's drug dependent or somebody who takes a lot of medication, you're going to see that their cells have a lot more smooth ER. They are a detox center. Remember that, okay? And the thing is you can actually tell somebody's level of resistance to drugs due to like the smooth ER. Smooth ER will build more and more smooth ER as you take more and more drugs, and that becomes your resistance, your tolerance to certain things. Like people can build up a tolerance to alcohol, but what's actually happening is at the cellular level, that smooth ER is like growing and making more smooth ER, and that's why it takes a long time, because it takes a long time to put that in there, it takes a long time to break it down too. So your tolerance for alcohol can remain for a very high level. So the key is not to build all that. You don't really want to have a high tolerance to drugs because, or alcohol, because it's not good for the body. Okay. All right. So moving on, that's our smooth and rough ER. Let's go on to the vesicles. Now, if you notice, I'm kind of skipping around this diagram, but going in order of that list. We did the nucleus, okay, I'm going to check that off. We did all the components of the nucleus. We talked about the ribosomes, both free ribosomes and bound ribosomes. We've talked about the ER, the smooth and the rough. So let's talk a little bit more about those vesicles. So we notice vesicles, and I don't think they're labeled on here, but they're certainly in here. There's vesicles that are entering this Golgi apparatus. This is all Golgi apparatus, which is labeled here. But there's also vesicles, here's some out here, that are actually leaving the Golgi apparatus and maybe even forming these things called lysosomes, okay? Vesicles are kind of like little transport globules that kind of bebop back and forth from the ER to the Golgi, from the Golgi out to become a lysosome. They're like kind of transferring all that protein. So we call them like little transports of proteins, little goblets. I kind of call them like little messengers going from one place to another. Okay, those are the vesicles. Now let's talk about that Golgi apparatus. I always say it's like a mailroom because it's going to sort, it's going to modify proteins, it's going to send proteins out. It's where the vesicles are entering and the vesicles are leaving to go to different places. And so the Golgi apparatus is quite a busy in and out center, I call it. In come the vesicles, out go the vesicles. But while that protein is in there, it's being sorted, packaged, and modified. In other words, like that folding, that primary, secondary, tertiary, quaternary folding, like the coils or the pleats and the globs on globs, that's all happening at the Golgi apparatus, okay? Continuing on, then, we've talked about vesicles and Golgi apparatus, are the lysosomes. So here's a lysosome. Like after some of these vesicles leave, these proteins within here might suddenly like take on a certain responsibility, and that would be digestion. Okay, some of the proteins in there become digestive enzymes, and they will be then considered a lysosomes. Lysosomes are such, again, another organelle that's like underappreciated. Lysosomes not only digest things like outside the cell, they may burst outside the cell and release digestive enzymes outside the cell, but they also may digest things inside the cell, especially organelles that are breaking down and old and run down. The lysosomes are going to do away with like the junk that's building up. They're almost like the custodians of our cells. They take out the trash. Now, an interesting disease called Tay-Sachs disease, it's common in the Jewish population, more common in the Jewish population, but common in any population, not common in any population. Terrible disease. What happens with someone with Tay-Sachs disease is their lysosomes actually don't function in removing lipids from brain tissue. This happens. A baby can be born perfectly normal, but as the lysosomes, they will be born perfectly normal, but as the lysosomes cease to function, lipids build up in the brain, and the brain is basically, smothered in these lipids. A child won't live usually past the age of 2 years old, and they will be very much deteriorating in that time. Tay-Sachs disease is an awful genetic disorder that a lot of geneticists are trying to help eliminate by eliminating that gene, going into in vitro fertilization, going into genetic counseling for a couple that might be carrying both recessive traits for that, because you do need 2 recessive traits to express that. So lysosomes, they clean out the junk. Okay, and a lot of times, when their job is going on at night when you're sleeping. That's when, especially your brain cells are being cleaned out. Your lysosomes are very actively busy and getting rid of all the junk. So lysosomes are kind of an important thing. All right, vacuoles. All right, I want to turn over to the plant cell to talk about. There's 3 types of vacuoles out there. There's more than 3 types, but generally speaking, when we look at vacuoles, we say they either contain food, they're either containing water, and they're a central large vacuole in plant cells, or they may be acting like a sump pump, like pumping water out, especially in one-celled organisms. So they can be food vacuoles, they can be contractile vacuoles, or they can be central vacuoles. Food vacuoles you're going to find mostly in your animal cells, okay? Contractile vacuoles you're going to find in a lot of one-celled organisms like the protists that live in like pond water. But the central vacuole, which is responsible for storing a lot of water for the plant for photosynthesis, you're going to find in plant cells. Okay, so that's your vacuoles. Mighty mitochondria. I'm glad we're on the side of the plant cell, because so often, and I don't think you guys get this way, but so often people think of mitochondria, and here's the mitochondria right now, as something that's only in animal cells. But no, they're in all cells, not prokaryotic cells, but they are in plant and animal cells. And the mitochondria, that's the site of cellular respiration. Mitochondria is also like a double membrane thing. It has an outer membrane, then the inner membrane has these folds called cristae. Deep within the inner membrane, you have a matrix, which has ribosomes in there. In between the membranes is that inner space. When we get into cellular respiration, we'll talk about, how electron transport chain, oxidative phosphorylation happens across the cristae, or happens across the membranes in the mitochondria. And that extra membrane space increases the surface area and allows for respiration to happen more efficiently. Okay, so that's the mitochondria. Plastids. I'm glad we're still on the plants. There are different plastids that they have. Now, the only one that they're showing, and if I can see if they're showing any more, no. They're only showing chloroplasts, right? And chloroplasts are, again, one of those double membranes. They have an outer and an inner, like the mitochondria does. But then their inner membrane can be sorted into these stacks of membranes. I always say they're like pennies when you stack them up, or pancakes when you stack them up. And the membrane around there then are called thylakoids. That's where a lot of the light reactions of photosynthesis are going to take place. Outside that, in the fluid, like outside the granite, is called the stroma, and that's where the dark reactions of photosynthesis are going to occur. The other plastids that are out there are leucoplasts and chromoplasts. A chromoplast may look very similar to a chloroplast, except it wouldn't have green. Chromoplasts can have any other colors, like your carotenes, which are orange, or your xanthophylls, which are yellow, or even your phycobilins, which are your blues and reds and purples, but they just won't have green. Green is like the #1 pigment, and when you find green, then most likely that's a chloroplast, because green is the one that's best at capturing the sunlight. Okay, so that's your chromoplast, would look just like a chloroplast. Leucoplast would look a little different. Even though they're labeling this as a peroxisome, a leucoplast might look very similar to a peroxisome, where they'd be clear and they're really just storing starch. Okay, so that's our 3 types of plastids: chloroplast, chromoplast, leucoplast. And if we're looking at the list, we're down here, we've done chloroplast, chromoplast, leucoplast. So let's talk about that peroxisome there. Okay, what is the peroxisome? Again, it's one that you don't learn in first-year bio, so this is a newcomer to AP Bio. Peroxisomes are another detoxifying thing, but how they detoxify is a little weird. They kind of like make a poison and then break it down right away. And that's what they're named after, the poison that they make. They literally turn substances into hydrogen peroxide. Hydrogen peroxide is actually toxic to our cells on the inside. So they quickly break that hydrogen peroxide down. And you all know that if you put hydrogen peroxide on a cut, you know how it bubbles right away? Well, that's as fast. As fast as they make the hydrogen peroxide, they break it down. And it breaks down to water and oxygen, which is like a beautiful thing. They take waste and they break it down into something. Well, they change it first into peroxide, hydrogen peroxide, H2O2, and then into water and oxygen, which is beneficial for the cell. So peroxisomes, a newcomer there. Cytoplasm, you know, that's all the colloidal suspension within, the fluid within. But in that cytoplasm is the cytoskeleton. I don't believe, oh yes, they are mentioning it. Thank you very much. Okay, they mentioned the 3 components of the cytoskeleton. I like to think of the microtubules. Tubules, they're little tubular-like things, like hollow-out tubes, but that's not actually where they get their name. They get their name from the protein that makes them, called tubulin. Intermediate filaments are made out of keratin, kind of like you think of your hair and keratin treatments. Okay, microfilaments are made out of actin. Actin, I always think action. Microfilaments are really remember the striations that we saw in the muscle cells? Those are all your microfilaments and all. And microfilaments are very important in movement within the cell. Intermediate filaments are a lot of times for anchorage within the cell. But your microtubules, they're like your bones that actually kind of give the microfilaments and the intermediate filaments something to kind of attach onto. And the microtubules are also responsible for structure within the centrioles and within cilia and flagella, and they're also responsible for when we pull those chromosomes apart in mitosis. Okay, so that was a quick look at all the components of the cytoskeleton. Cytoskeleton, again, it's a supportive and movement network within the cytoplasm. And it was only discovered, I believe, late 80s that they started being able to see it under a microscope, and they were like, oh, what is this and what's going on here? So it became much more important. Okay, we got to flip over the sheet because in animal cells is the only place we find these things called the centrosome, which is made up of 2 centrioles, okay? And here it says centrosome. It's actually this structure within here. And you would find 2 centrioles. Here's one at this angle and one at this angle. There's always 2 of them inside a centriole, and they're at right angles to each other. And they're these little tubular things, little My slinky kind of like is a good one. If I had another slinky, putting it next to it there, that's another good one. Centrioles are going to be responsible for cell division, pulling those chromosomes apart. But the centrosome itself, that whole area, really organizes microtubules. Microtubules from the cytoskeleton, as well as microtubules that will be responsible for pulling the chromosomes apart during cell division. Okay. Cilia and flagella. Well, we have a flagellum on this, and we do have microvilli. They're not the same as cilia, similar, but microvilli are a little bit bigger, okay? Microvilli are just extensions of the cell membrane, which kind of cilia is too. Let's see if there's any cilia over here on the plant. Didn't think so. So cilia and flagella are for movement of the whole cell or movement of things past the cell. Flagella, obviously, sperm cells have a flagella. Gets the sperm to its destination. Some protists have flagella. Some bacteria have flagella. They're really moving the cell to its destination. Cilia, on the other hand, which would be finger-like projections, tinier than microvilli, on the surface, but are little extensions of the cytoplasm kind of poking through the cell membrane, are responsible for things like in your lungs and in your nose, your whole respiratory system, for moving fluids and dust out of, and mucus out of your lungs. Cilia can also be found on unicellular organisms to move it around. Cilia, most of the time in our cells, are brushing things past the cell, but could move the whole cell. Okay, and by the way, just to get you all excited, because this video is going on now for 21 minutes, holy cow, we're done all those things. So we're down to the nitty-gritty here, and the last few things. And first there's the extracellular matrix. Well, fortunately, I gave you an ed puzzle, so we're not going to talk about that too much. But I like to just mention the extracellular matrix. Usually we're talking about animal cells when we talk about extracellular matrix. Is just a collection of carbohydrates and proteins that really kind of bind one cell together to another cell and make the tissue. It's made up of collagen, these proteoglycans and fibronectins that all like kind of connect one cell to the next. Animal cells usually. Cell wall. Got to flip it over, okay? And the cell wall of plants. Now, what's interesting is if you're looking just at this plant, you see the cell wall all around, right? And then here would be the next plant's cell wall. Okay, cell walls are made out of cellulose. We can't digest cellulose. So anything that has thick cell walls is going to come out of your digestive system going in like it did. You don't digest it. That's fiber in our diet, and it's good. It scrapes all the stuff out of our digestive system and cleans our poopies out of there. But in the cell wall, and you can notice these little tunnels here, that's the plasmodesmata. They're like little things that connect one plant cell to the next and can transport fluids. So when water transports up the stem of a plant, or up the roots of a plant to the stem, to the leaves, it's going through these little plasmodesmata. That's how it goes. Otherwise, it wouldn't be able to seep from one cell to the next. A lot of people think, oh yeah, the cell can just pass it. Well, how? It passes actually through these little tunnels called plasmodesmata. Now, in an animal cell, and it's not going to show this too much, you have to really look at a diagram in your book to show you all the like junctions that they have. In animal cells, I'm going to bring up the list here of structures that join animal cells together, are these three things: the tight junctions, the desmosomes, and the gap junctions. Tight junctions are just like they say. It's where like two cell membranes tightly connect together and may have some protein fibers through them that actually help them connect. These are really important for leakage, okay? You don't want your intestines like leaking into your body cavity. You don't want your skin leaking out substances. We can thank tight junctions for that. Okay, tight junctions between our skin cells keep like fluids from exiting our body too much, holding us together. And they also keep like our digestive enzymes all within our intestines as opposed to getting into our body. Now, desmosomes are tight rivets. You could have leakage around them, but where they are together are really tight. And this is in muscles. So like muscles could leak from one muscle cell to the next cell, which is not a big deal, but you don't want those muscles tearing. In fact, when you do have a torn muscle, it's usually the desmosomes that you've torn, those rivets. And they're very powerful, but they're tiny, and they're really important in muscles, connecting muscles together. And finally, gap junctions. Just like the plasmodesmata, animal cells have gap junctions, which are little tunnels from one cell to the next cell. And that does it for all our cell functions. That's a lot in the eukaryotic cells. I wanted to do this video for you to just listen. I'm going to give you a copy of these cell structures and functions. I'm going to attach that to it as well. I'm going to, you already have your cell diagram. If you remember your cell diagram that you did that had all the parts and functions. Here's mine. Here's actually the answers on the back. But I did grade yours, so you should be able to see that. You've got an animal cell up here and a plant cell down here. So you should know all these parts and functions, but we did go into a lot of detail about these parts. Please, 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 I don't want to quiz you on this stuff. But it's not as simple as we learn in first year bio. It's not like the mitochondria is the powerhouse of the cell. We got to get over that, and we got to say it's the site of cellular respiration. Yes, cellular respiration, the production of energy. That's why they call it the powerhouse of the cell. But we need to be a little bit more detailed oriented when it comes to our cell parts and functions in AP Bio, okay? So pay attention to this, look this over, hold on to this diagram, it's important, okay? And we're going to go into details of the cell membrane. That's what 2 of the Ed puzzles have been on already. And then talk about transport from one cell to the next, good old diffusion osmosis, all right? And that's it, you guys. I know it was long. Thanks for listening.